Well, to that end, the grades have been posted. Any questions, concerns, please reach out. Happy to talk to everybody about them. And I want to offer reassurance, anyone who's concerned about how you're doing, nothing is out of anyone's hands by any grades that I've seen. We still have the vast majority of points to go. But if you are concerned, or if the first exam did not go well, please reach out as soon as possible. And as we just heard, you can reach out to me, or we're setting up mentors within the Farm Tox Club, so there will also be student mentors there. Yes, please.
Do you know the average of the exam, like the score average?
It was roughly 80%, about an 80-ish, so that would have been 24 points, 24 out of 30. Yeah, average. I usually don't post all that. But, yes, please.
Did you curve the exam at all?
No, it's a straight scale, but there usually are points that come back at the end of the semester as you go through the exams, and that certain questions will be cast into doubt and they'll get booted, so there could be points coming back. So no curve for the class, but final point total usually adjusts a bit at the end. Other questions or concerns, anyone? Good. Okay, so, right, and then we've got a quiz coming up Thursday. There's practice exam on today's module that you can take over the next couple of days. Today's going to be the final part of alcohol lectures, and then get into other CNS depressants. We'll see in a minute. However, before I dive into that, I believe I told some people I was going to post this before the last exam, and I apologize. I have this very generic advice for how to study for any of the classes I do, so I wanted to put it in here just for what it's worth. But the usual steps I recommend for studying for the class, the first thing you have to do is get vocabulary down. Go through every lecture in whatever section for that exam or quiz or whatever you're studying, and chart out all vocab. And then learn them, anything that you're unfamiliar with, learn them in your way. Flashcards, writing it out 10 times, however you do it. Learn it so that as soon as you see the words, you know exactly what it means and you don't struggle to stitch those concepts together. That's the first crucial part. Once you have all of the terms down, then go through each lecture and go from biggest to smallest in terms of concepts. Obviously, for the upcoming lectures, each one or a couple lectures will be based off of one drug or group of drugs. So that's a good place to start. And then chart them down into the smallest detail possible for that. Then after that, that should be 2 full times through the notes, where you have things outlined and you've been through them enough so that they should be familiar. That means the areas or things that are unfamiliar will really stand out, and those you go after then. Reach out to me, reach out to potential tutor in the Farm Tox, someone else that you know that's in the class or has had the class. However, reach out to someone in the know and get help with that material. That's 3 times through the material, and I hope that should be enough. But anyways, that's how I usually recommend to study. If it helps, for what it's worth. But questions on that, studying, exam stuff, grades. All right, please let me know if you do. So let's dive in here. To start off in a very broad manner for today's lecture, going back to our DSM-5 manual, if you have any 3 of the following 7 symptoms, you are known as someone who suffers from alcohol use disorder. This also applies to any addictive drug, really. You can substitute alcohol in here with your drug of choice, but developing any of these symptoms, tolerance, withdrawal symptoms, or using alcohol to relieve withdrawal, drinking more than intended, unsuccessful attempts to cut down on use, excessive time related to alcohol. If you're cutting work or class so you can go by, that's a big no-no. Spending a lot of time being hungover doing your normal activities. impaired activities, professional and social, as well as continuing to use even though it will affect your life in a negative way. Any three of these mean that you are probably subject to alcohol use disorder, but this is a good list to learn for the future, or for upcoming exams. So, with that in mind, getting back to alcohol, or the molecular aspects of alcohol, specifically pharmacodynamics of alcohol, we talked about this a bit last time. The genetics of alcoholism, so I mentioned alcohol and aldehyde dehydrogenase, dopamine receptors, and now we can throw GABA A into the mix, right? So GABA A is the distinct target of alcohol. Here's a structure of the receptor here, so ligand-gated ion channel, membrane here, or cell membrane here, inside the cell, outside the cell. You don't need to know this exact structure. Knowing just a bit about the basic components will be important for later, but I'll go into that in what is important. What specifically you need to know are that this is comprised of subunits. So this is a protein, technically a pentamer, so it's a complex of proteins that 5 individual proteins make up and form into this channel that then embeds in the membrane. We'll go into some of the details in just a minute, but as this shows, we saw last time, there are distinct areas that molecules or ligands will sit in this receptor. GABA, so here's the endogenous ligand right here, binding to the outside of the receptor. Alcohol is down in here within the channel, and then barbiturates and benzodiazepines bind at different areas, but we'll get into those today. Talked about picrotoxin, that's an antagonist, so it'll keep it from activating. Yes, but last thing I'll say about that, there are also things known as endophenotypes that are linked to alcoholism. And these are more groups of genes that are passed down through your genetic lineage to you, and it might not be just a specific gene for a phenotype like hair color or eye color or the length of your intestines or something like that. But these are groups of genes that carry through distinct genetic lineages that give rise to distinct behavior patterns. In this instance, endophenotypes towards alcoholism is another type of genetic predisposition towards this usage disorder. But it's not just a specific gene or mutation of a gene, these are groups of genes. So endophenotypes can also lead to predisposition for alcoholism. Okay. So, last time we talked about mechanism of action for alcohol revolves around GABA A receptor activity. That's one form. That's the initial inebriation and decrease in anxiety that we feel. However, with long-term use, there is activation of the reward circuitry in the brain, and we'll deal with that now. So this is alternative mechanism of action for alcohol. So we, just a quick aside, again, if the first exam didn't go well, or if the material, you struggled with that material, this is a great figure to commit to memory or to learn very quickly, learn very thoroughly right now, as this is going to come up again and again and again in most all of the drugs that we're going to talk about. But let's get oriented here. We're going to spend a little bit of time with this figure. So, cross-section of a brain, ventral tegmental area, nucleus accumbens, and PFC, all right? So VTA is here, nucleus accumbens here, and prefrontal cortex here. We saw this last time. We have an interneuron within the VTA of GABA, then we have a projection neuron. This time we've got dopamine going out, synapsing in the nucleus accumbens and the prefrontal cortex with dopamine neurons in both of those areas, all right? This whole thing revolves around this dopamine neuron right here, okay? We're gonna start right with that neuron there. Obviously, in reality, it's not a single neuron, these are groups of neurons in this area, but the whole field of addiction really is built off of dopamine, right? I mean, it's all about dopamine or dopaminergic tone in our brains that will set us up for addiction. This is the neurotransmitter that really says, something amazing is happening, I need more of this, I want to pay attention to this. That's the most bland way you could state this. Because within the field of dopamine research, there's a couple of camps that oppose each other's view on what dopamine really does, and that's outside the scope of this class, but the bottom line is addiction is still revolves around dopamine tone, okay? Back to this. So we've got our dopaminergic projection neuron in the VTA. This will synapse in the nucleus accumbens and the prefrontal cortex, releasing dopamine onto these two neurons that can then be activated and do their own thing in those two areas, all right? Under normal situation, the dopaminergic neuron here in the VTA is under the control of this GABA interneuron, right? GABA is on, that means dopamine is off, right? GABA is our inhibitory neuron. When that guy is on, that's the normal state of things in the brain. GABA will be on, dopamine will be off, all right? Now, what we saw last time is that we can inhibit this GABA neuron and allow for activity to happen within the brain. However, more closely to related to what we're talking about, alcohol's effect can also be imposed on the hypothalamus that causes the pituitary production of an endogenous opiate molecule, beta endorphin, and then that in turn can come back and act on its target receptor, the mu opioid receptor, on this GABAergic neuron, okay? So what happens here is we consume alcohol, it affects the hypothalamus, that triggers production of beta endorphin by the pituitary, that's released into circulation, and once it gets into this area of the VTA, it can bind to mu opioid receptor and shut off GABA, okay? So we've got GABA is now off, dopamine is now on. That dopamine can then turn on dopaminergic neurons in the nucleus accumbens and PFC, and that's the start of our reward pathway, okay? How does that sound? Just hold, just with thumbs up. Okay, so just for Department of Redundancy Department's sake, dopaminergic neuron in the VTA projects and synapses in the nucleus accumbens and PFC. This is our reward pathway right here. We're going to get more into that as things go on, but this is the base of it. When we consume alcohol, it can trigger the hypothalamus to cause the pituitary to produce beta endorphin that binds to mu opioid receptor on the GABAergic neuron in the VTA, shutting it off. When GABA is off, dopamine is on. GABA is on, dopamine is off, right? Okay. So, moving right along. As far as alcohol toxicity goes, a lot of it revolves around our liver, for the most part, but there are several different aspects to it that, well, let's just walk through this and then we'll get what's important. So, chronic alcohol consumption causes increase in alcohol dehydrogenase, CYP450. Which CYP450 is for alcohol? 2E1, good job. And then also it can cause problems in the GI tract. So overexpression of these enzymes can cause imbalance in things, especially reactive oxygen species. So free radicals can be generated. And those can cause damage to any biologic tissue, really. However, imbalance in free radical metabolite production, as well as actual destruction of the GI tract through repeated exposure to alcohol itself can cause intestinal permeability, and then the bacteria in the gut can leak out and cause all kinds of problems. As you can see, these different pathways lead always to liver inflammation. Long-term liver inflammation can lead to necrosis, and then body tries to respond through fibrotic or development of fibrotic tissue, but this ultimately leads to cirrhosis and really your liver just shutting down. So, without needing to understand all of the steps in this pathway, what's really important is that chronic long-term ethanol consumption leads to inflammation in the liver. Long-term situation there leads to development of fatty liver, then inflammation, and then cirrhosis. And then a couple of other things that can happen. Wernicke's encephalopathy is a B1 deficiency that happens when you have an improperly functioning liver. Well, a cirrhotic liver, it's not just improperly functioning. This can cause confusion, ataxia, visual disturbances, and that can be rescued with treatment of B1, but usually by this stage you've got a multitude of other problems going on. But this can also lead to Korsakoff psychosis, and this is an actual loss of neurons in the brain. So, and this is a fairly rare one. You really have to hit it hard for a long time to cause the kind of damage to a brain. But yeah, so Korsakoff psychosis, Werner's encephalopathy, bad conditions that happen with long-term alcohol poisoning are the result of alcohol toxicity. Couple of last notes on alcohol. So treatment we talked about last time, disulfiram. This actually inhibits aldehyde dehydrogenase, remember, so our ethanol metabolism is like this. Here's our ethanol. It can be broken down by CYP450, 2E1, or alcohol dehydrogenase to produce acetaldehyde. That molecule then gets broken down by aldehyde dehydrogenase to produce acetic acid. And disulfiram blocks ALDH or aldehyde dehydrogenase. And it's used as treatment for alcoholism, but as mentioned last time, the biggest problem with this is compliance, because nobody wants to take it that has been given this drug, because it just, yeah, you take it, you inhibit your ALDH, your body, if you drink, your body produces a whole bunch of acetaldehyde and it just makes you sick and miserable, and it's not fun, so nobody wants to take it, but I mean, it will curve you from wanting to use alcohol for sure, but unfortunately most people just stop taking the disulfiram and keep drinking. Is the biggest thing they found. Two other drugs that are used for dealing with alcoholism. Naltrexone, so this is an opioid receptor antagonist. So looking at the what we just checked out in terms of how alcohol actually triggers the reward pathway and causes those feelings, or starts us down the pathway towards addiction. And by triggering production of beta endorphin, and then it reacts at its target opioid receptor. So naltrexone acting as an antagonist will actually block this. So the idea is that you can still get some pleasant feelings from using alcohol, just due to the activation of GABA A receptors. However, the thought is that with naltrexone on board, you don't develop the increased dopaminergic tone in the brain, and you don't develop that sort of addiction to that new elevated level of dopamine. So it blocks this from having an effect. So you could still have some good feelings, but the idea is that you don't have the reinforcement in your brain, and you lose that sort of drive to have alcohol present, which is common in alcohol use disorder. Acamprosate is another one that, this is more for dealing with people who are right at the first steps of recovery after long-term alcoholic use. So we talked last time about what happens in the body when we have alcohol present chronically, and we showed that it'll shut down that glutamate release from the glutamatergic neurons in the brain, and well, in the amygdala, and then what happens is if we keep exposing that system to chronic ethanol presence, the body will try and respond by upregulating calcium channels, upregulating NMDA receptors, producing a lot more synaptic vesicles with glutamate in it. And then all of a sudden, if we take the alcohol away, everything opens up and it just spills out. We talk about delirium tremors due to the fact we have this overabundance of glutamate signaling going on in the brain. You get the shakes and it's real bad. That's what a compressate is meant to deal with. It's specifically an NMDA receptor antagonist, so the thought is that it'll stop you from shaking and having those sorts of side effects during your withdrawal phase, okay? So some of the really bad effects that happen to the most extreme cases, this can calm them down. But what most commonly happens is you just get a script for what we're going to talk about next, and you just go on benzodiazepines, and it'll just mellow you out completely. So a compressate is still used in extreme cases, but it's not that popular of a tool anymore. But I believe that's it for yes. Alcohol. Questions, comments? Yes, please.
So will there be any dysfunction in the regular use of the GABA neurons due to naltrexone?
I'm sorry, would you ask that again, please?
Will there be any change in the way of using of the normal GABA neurons, like the normal release of dopamine, if naltrexone is introduced?
Okay, I think, good question, yeah. So is there any affecting other GABA neurons outside of the VTA that, so it can if they have a mu opioid receptor on, okay? So you can affect other GABA neurons elsewhere, but they would have to have the specific receptors, and they don't always. You know, those are kind of specific to VTA GABA neurons. So, but good question. Yes, please.
So like going off of that, like, cause obviously dopamine has like a really big role in our emotions and happiness and things. The person that is on that drug, would they still be able to like regulate those emotions, or does that seem like being harder during that time of pain?
Gotcha. So yeah, with the effect of the dopaminergic neurons in these areas specifically, will that spill over into other areas of the brain? Yes, it can, but it's usually more sovereign to this because, yeah, we cover very limited amount of control for what our brains really have, right? So there's a lot more control structures involved in the brain that keep things better regulated. Yeah, to answer the question, yes, there can be spillover with other areas, as we are definitely starting to uncover now how addiction is being seen more in terms of outside of just its physical and psychological presence. The emotional sphere of what people go through in there is now being more appreciated, and so that would definitely come into play with dopaminergic neurons outside of these areas. However, the amount of control that actually happens there allows allows the situation to not have as much spillover, and it really, yeah, allows for just these pathways to be activated or inactivated. So that helps. Yes, please.
Can you go over the disulfiram mechanism one more time?
Absolutely. So disulfiram, we've got alcohol breakdown happens in 2 steps. First step is either P450 or ADH, right? The product of either of those pathways is aldehyde, acetaldehyde specifically, all right? Next step, acetaldehyde is broken down by aldehyde dehydrogenase, and that is what disulfiram inhibits. So it's specifically targeting that because of the fact that the byproduct acetaldehyde is toxic to our system. And this was developed decades ago when we had a much different kind of perspective on pain and punishment as we do today. So it was developed because it is such an inconvenient therapeutic and it causes people to undergo such unpleasantness. But that's the mechanism of action. Disulfiram triggers or inhibits ALDH, and that's why it's used for alcohol addiction to curb that.
Thank you.
Very well. Yes, please.
So this also crosses the blood-brain barrier.
The disulfiram, that's out in periphery.
Okay.
This is in the liver. Yeah. So, no.
Across the blood-brain barrier.
Disulfiram, that's a good question. I don't know if it does get across the blood-brain barrier, because the other thing is there's no narcotic effect with disulfiram, so you can take it and it really doesn't do, it'll inhibit this enzyme. But it doesn't, yeah, I don't know of any target in the brain that it would go, it wouldn't target anything in the brain. So, yeah, what we're looking at here is happening in the liver. And yeah, I don't know if disulfiram would cross the blood-brain barrier, but I don't know what it would do because there's no target for it there. Or there's no narcotic effect, so. Yeah. Other questions, please?
Okay, so when I was in high school in my health class, so being counts out drugs and pills, alcohol kills neurons, and then I learned today that it's only in extreme cases. Is that what they were referring to, or is that just used as like a tactic to like try and like purpose like discard alcohol use?
I don't know exactly, but yeah, alcohol definitely does kill neurons. And however, though, that statement sounds like a cell culture kind of thing. If you grow nerves in a dish in culture and you dose them with low concentrations of ethanol, you'll see inhibition and death and all kinds of bad stuff there. There's enough buffering in our body in that you don't have that extreme of an effect, but it is, alcohol molecule itself is toxic to biological tissue. So there's that, but usually there's enough buffering between drinking and it getting into toxic level or actually killing neurons that it's not as dangerous as what you would see in a lab, so.
Okay, so it might have been rhetoric to like discard alcohol use on high schoolers.
Absolutely is, yes, and for understandable reasons. I don't mean to downplay that or just say that's just some lab thing you can show anything with, but yeah, there's definite toxicity of alcohol with neurons and any biologic tissue, and I don't know exactly what the motivations were that what you heard, but there's a lot of buffering that happens in between consumption of alcohol and then actual neurotoxicity, so. Yeah. All right, how we doing, everyone? I'm bored with alcohol. Smiles over there. Not. All right, let's move on. So, as we'll talk about in the opioid section, that's been something that's been around for thousands of years. Opioids have been used as a sedative and anxiolytic as well as analgesic for a long, long time. But upon isolation of the active molecules and then production of high concentration opiates, we immediately saw problems with that. Again, we'll get into that in that section. But come back to today's lecture. When the medical community was seeing the problems with opioids, they wanted to develop things that could be used as analgesics or anti-anxiety medications that didn't have the side effects of opioids, all right? So, one of the first series of molecules that came about, this is really the base structure here, melanolurea, or barbituric acid. So this is a scaffold that a lot of barbiturate molecules were built off of. And yeah, this is background stuff. So this is the background slide on barbiturates. Don't worry about all these names. Definitely structures you won't have to know. I just threw them up there for people who like that kind of stuff. But the bottom line is, the first generation of molecules that were developed to move into the space of sedatives, anti-anxiolytic, or anxiolytics, not anti-anxiolytic, anxiolytics as well as analgesics were barbiturates, all right? And these had tremendous effect, but immediate problems. So here's some more structures here. Again, you don't have to know the structures, and I list the names just for background. Pentobarbital and phenobarbital are still the most prominently used in a clinical setting, though not so popular anymore, and it's really due to their abuse liability. So the way that these work is similar to alcohol in that it will potentiate the effect of GABA, okay? So barbiturates act on the GABA A receptor, and cause a membrane potential to go even more electronegative. So if we're looking, I'll go a little bit more into this in the next slide, but if you have a cell that is expressing the GABA A receptor, and then you treat it with something, its own endogenous ligand, GABA, what you see, so here's a resting membrane potential at negative 60 millivolts. You treat it with GABA, you get it to go more electronegative. Here the example is showing it moving to negative 60 millivolts, but if you put the same amount of GABA with well, this is pentobarbital, but a barbiturate, you can see a massive decrease in the electronegativity, meaning it's harder for, so our action potential would be up here, right? And it has to pass that threshold level, so it has to increase the positivity in the membrane. And once it gets to a threshold level, it'll trigger the actual action potential. So that value would be up here. So you can imagine the farther away from that you get would be harder for an action potential to happen. Yes, please.
Does it only, does it do anything on its own, or does it only help GABA
So
respond?
yes, it can work on its own, but it won't commonly because there's enough GABA around. So yeah, will it work on its own? It can bind to the receptor on its own. But in situ or in a real life situation, there's going to be GABA around, so it's going to potentiate that. So, yeah.
Well, thank you.
Please.
Is this suppression why phenobarbital is used for seizures?
That, yes, is the suppression why this is used for seizure. That's exactly right. As they, overlap here will be that the thought is that you have way too many of these action potentials happening without coordination that usually does happen. If you suppress them all, you don't have to worry about coordinating them, and that is why this is used for seizures, among other things. All right, so yeah, this is kind of the same thing, just in more real data. And to go over, so if anybody has done electrophysiology before, specifically with, expressing receptor in oocytes or sort of single cell setups. So you express a receptor, and yeah, we don't really have to go through this, but for those who are interested, you can express a specific gene in a single cell type of system, and then you record the membrane potential of that if you dump on different drugs onto that single cell. And then you can get readings like you see below here. So again, here's our example from the previous slide. And then if you look here, these are 1, 2, 3, 4, 5 different traces of the same kind of repeat situations, all right? And if you look just at this line here, C stands for close, O stands for open. So the amount of time that you see this trace here, this is a channel open, this is channel closed. And what you should see between these two, because this is pretty muddy data, but still, I don't know, it makes a point. What these drugs do, not only do they potentiate the activity of GABA, they will allow for a longer opening and a longer opening for that specific channel. So if you see here, this is different setups being treated with just the endogenous ligand, and you can see the majority of opening here. Compare that to over here where we see a lot more time with channel open, and you can even see instances where you have a magnified effect, if you will, where you have a certain amount, and then outside of that, and this is all about receptor occupancy, et cetera, but just the bottom line here is that with pentobarbital on board, you have a lot more channel opening for a longer duration of time compared to just endogenous GABA ligand, okay? So, bottom line, barbiturates target GABA A receptor, causing a longer amount of open time versus just GABA alone. So, one of the problems with them is that these have a very narrow therapeutic index, right? So here is yet another version of looking at the difference between a therapeutic dose and a toxic dose, okay? So these three plots, we've got phenobarbital this time, diazepam, and alcohol. So all three CNS depressants. Alcohol we know and love, diazepam we'll get to. These are benzodiazepine, or this is kind of the quintessential example of benzodiazepine. And then phenobarbital, the barbiturate here. We're looking at the effects of the drug plotted as a function of the concentration of drug in blood, all right? And what we see is we have the anxiolytic effects, so these are the anti-anxiety effects of these drugs. This is our therapeutic dose, right? So the lower doses, that's what we're looking for. We're looking to relieve anxiety with these drugs. However, at a higher dose, we're getting sedation, okay? Sedation can come in the form of just going to sleep. It can come in the form of going to sleep for good. So sedation is our toxic level of drug. And as you can see, there's a different span between our anxiolytic and our sedative doses. Alcohol and benzodiazepine is fairly wide. Barbiturates is very narrow. This is one of the immediate problems they had with these drugs, is they were having people overdose really easily. And the problem is they just feel so good, you just keep taking them, and then you go to sleep, and that's it. Okay, so cross tolerance happens between these drugs and alcohol, so be forewarned about that. If you take barbiturates or benzodiazepines or alcohol, they can cause cross tolerance with each other because they're all targeting the same receptor, all right? Here's that receptor picture again. I'm not exactly sure why I put that in there, but we've got anxiety effect plotted as a function of dose of barbiturate. You will need less of it with someone who's not used to drinking alcohol than you would with someone who is experienced drinking alcohol. So you need higher doses because it right shifts our potency curve, meaning you need more of the drug. And these you can mix and match between alcohol, barbiturates, and benzodiazepines, okay? But just remember that they do have this cross tolerance within this group of molecules. So then, medicinal chemists went back to work because not only do barbiturates have a very narrow therapeutic index, they developed tolerance very easily, and the biggest thing was abuse liability. People were getting addicted to these immediately, and as I was saying initially, they tried to develop this medicine as a cure for opioid addiction. And then sure enough, people became addicted to barbiturates. So the medicinal chemists came up with benzodiazepines. Initially, Librium was the first molecule that was synthesized, but diazepam or Valium is definitely the kind of quintessential molecule now. Rohypnol, I'm sure people have heard of roofies before. That's an awful drug, but it was developed mostly for not just anti-anxiety, but the sleeplessness, for insomnia. However, there's spillover with memory, and people dosed with this tend to not to be able to remember things as well as being inhibited or being sedated. And then Xanax is one a lot of people use for relaxation, but also for more social, I guess, applications. However, so these were benzodiazepines. These were formed to deal with the issues with barbiturates, so they're safer. They have a much broader therapeutic index. They do develop tolerance. It's not as bad as barbiturates. However, they still have an abuse liability because these also make you feel good before they make you pass out. So people started getting addicted to these. However, the abuse potential isn't as bad as it is with barbiturates. But There are continued studies to make even a third generation and fourth generation of these BDZs that are getting much better, and we'll get to that right at the end of this, but these are examples of benzodiazepines. Typical and atypical benzodiazepines. These are the only two atypical benzodiazepines. So Zolpidem, Izapiclone, or Ambien and Lunesta as they're better known. These really were developed as insomnia medications, okay? They do have anxiolytic properties, but they were really developed to help people get to sleep. And yeah, there's definite abuse liability with these, but just remember these two atypical benzodiazepines were developed for treating insomnia, not the other host of things that other BDZs can target. Okay. All right, so I got just a couple of, I don't know, outlier medicines that fall within this space that I'll cover quickly. GHB, gamma hydroxybutyrate, similar to GABA, as you can see the structure here, just without that amine for GHB. And this is a really interesting one because it has some very distinct clinical benefit, but then also horrible abuse potential due to the fact that it has a very short half-life, so you can take it and then it disappears really quickly, but it also produces amnesia, and this is how unfortunately it is used in a very nefarious way because people who are subjected to this have a problem functioning and then is remembering what happened to them. But just to go over how it works, actually works through GABA A receptor as well. GHB can bind, or it has potency at GABA A and GABA B. However, GHB can be converted to GABA to bind GABA A as well, but it also has its own receptor, GHB receptor, and this also can trigger ion channels and have a similar effect as GABA in the body, all right? But due to the fact that it has more widespread target receptors than just GABA, this is why it causes the problem that it does and triggers the problem with amnesia and memory loss. But just remember, GHB can target its own receptor as well as GABA B and GABA A. But that, propofol, so technically an anesthetic, it's used in primary induction anesthetics, if you know, yeah, the clinicians here, propofol, ketamine, drop the third one, well done. But these are used for putting people down and then, or putting people to sleep, and then longer term anesthetics can be used there. But as just mentioned, this is a powerful sedative, however, thought to be very safe due to the fact that it can clear very quickly, and when used under clinical supervision, it's very safe. But yeah, puts people to sleep quickly, its effects wear off quickly, but then with a properly trained anesthesiologist on board, they can maintain that sleep for surgical procedures and whatnot. So, got us there. Yeah, okay. So now we're going to go into some in vivo studies. Questions on what and whatnot, whatever I just covered? Yes, please.
Is there a potential for abuse for propofol as well?
Is there a potential for abuse for propofol? Not that I'm aware of, because it doesn't really, it shorts you out. So yeah, its potency is so high that it will, you kind of skip over the fun parts of the going to sleep. It just puts you down. So I can imagine a situation that there is, but for our purposes, let's just say that there really isn't a abuse potential for that, and it really just has to do with that. It, yeah, knocks you out quickly, and you really don't get any narcotic or any of the goods. So, yeah. Okay, so when talking about measuring anxiety in animals, rodents, because these are really designed for rodents. The elevated plus maze we talked about before, and novelty suppressed feeding. Novelty suppressed feeding really plays on a mouse's desire for protection. So if you drop a mouse into an open area like this, it's still confined as a little cage or a little tub or bin, whatever you want to call it. It will spend all of its time out here on the walls, walking around, because that's where it feels most protected. It'll spend very little time going outside, okay? Even if, as you see in this instance, you have a little disc here with food on it, okay? Let's get into that in a second. So, elevated plus maze we talked about before, so this is a plus shaped maze. You have 2 arms that are covered, 2 arms that are open. If a mouse is traveling out in this open area, they're thought to experience anxiety. In the closed area, they're not. So if a mouse is undergoing anxiety, it will stay in these areas. If it's feeling more calm, it'll venture out into the open arm areas, and that's how you can measure the anxiolytic effect of a drug. Same with novelty suppressed feeding. A naive mouse will eventually leave the comfort of the wall and come out here and get food, but you can measure the time it takes to go from that brave moment when it breaks away out into the feeding area and show a decrease in the time it takes when you were experiencing less anxiety, meaning you have a drug that is an anxiolytic. So, for the elevated plus maze, again, time in the closed arm is for, if you're experiencing anxiety, you want to spend as little time out in the open arms as possible. So looking at the left plot here, we're talking time spent in these arms, closed arm, a lot of time, open arm, a little bit of time, just its natural exploration, but this is no drug treatment. So this is normal behavior of a mouse. It'll spend most of its time in the closed arm, very little time in the open arm, because it is elevated as well. So it can look over the side and look down, and it's like a lot of people, a lot of animals, they have a fear of heights. However, throw a nice strong anxiolytic in there like Valium, and it will mute that effect, meaning the animal will not experience anxiety and will just roam around in the plus maze, checking everything out. So it it equalizes the amount of time spent in the closed and open arm. It doesn't feel the anxiety that it usually does in the open arm, so it's willing to travel out there an equal amount of time, okay? So this is a standard in vivo experiment for monitoring the anxiolytic activity of a drug. Okay, so, and similar, the novelty suppressed feeding, again, throw the mouse, gently place a mouse into this enclosure, and it'll spend its time running around the outside before it dares to venture out into the middle. If you give it a nice dose of Valium, it doesn't care if there's an eagle in the sky. It goes right out there and it goes for that food. So, these are 2 ways of assessing the ability for a drug to decrease anxiety in a rodent. Elevated plus maze, novelty suppressed feeding. Elevated plus maze, they'll spend an equal amount of time if they have an anxiolytic on board. Novelty suppressed feeding, with no drug, it takes them a long time to get off of that wall and get that food. If they've got benzodiazepine, alcohol, barbiturate, it's going to go right out there and start chewing down. Okay, back to our GABA receptor. So I keep showing this because this is made of different subunits. Those subunits are alpha, beta, gamma, some combination, and then within each of those letters, there are different subsets or subunits within the subunit, and it can be a combination, or this pentamer is made up of 5 of any combination of these different subunits. Here is an example of the most popular of them. You don't need to know that for an exam, it's just background stuff, but what I would like you to know is where these drugs bind between the individual subunits, and that's shown right here in this diagram. So GABA, the endogenous ligand, binds between alpha and beta subunits. Barbiturates bind to beta subunits. Benzodiazepines bind between alpha and GABA subunits. I believe I told someone you don't have to worry about receptor structure, stuff like that. I apologize if that was misleading by saying what I'm saying now, but you don't have to worry about the structure and all of that. But I would like you to know the individual subunits, just alpha, beta, gamma, don't need to know all these particulars here. And know that GABA binds between alpha and beta, barbiturates bind on beta, benzodiazepines bind between alpha and gamma. That's all you need to know, okay? And the reason I set all of that up is because when talking about developing next generation molecules for benzodiazepines, they're the best that they've been, and of course, yeah, they have the best therapeutic index that we've seen so far, lowest abuse potential that we've seen so far, and they've been able to really develop them so they'll just treat insomnia, or they'll just treat anxiety, or they'll just provide some sedation for cases like seizures. Okay, now this top chart just shows a bunch of outputs from the amygdala. So they have inputs from these areas of the brain into the amygdala, and then outputs out here. Not so concerned about any of this. Why I want to show this is, as you can see from this, so this is a brain scan of a young fetus. And what they're highlighting is the expression of alpha-1 and alpha-2 subunits of the GABA-A receptor, okay? So areas that are really light here, 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 they have the highest amount of alpha-1, and then down here, the highest amount of alpha-2. Alpha-1 GABA-A subunits are known to be involved in anticonvulsant and sedation activities. Alpha-2 are more for anxiolytics, okay? So if you want to develop a drug that just treats anxiety, you would want something to target alpha-2 subunits of the GABA-A receptor. And here you can see there's where the cross-section is red or white, that's where there's a lot of expression. So in these areas here, if we go up here, you don't see much alpha-1 here, right? Alpha-1 is predominant in these areas, alpha-2 here. Alpha-2 is for anxiety, alpha-1 for the sedation and seizure or anticonvulsant treatment. So, just remember those two things and you should be fine. That's it for this section. I believe we got cannabinoids coming up. Big one on Thursday.
What are we